Hi, my name is Matt Walter. I'm a physician scientist at Washington University in St. Louis. I specialize in the care of patients with MDS and have a laboratory that studies the genetic basis of this disease. I'm also the director of the Edward P. Evans Center for MDS at Washington University. So I was fortunate to receive funding from AAMDS in 2005 when I was just starting my laboratory. And that research was focused on understanding the DNA mutations that occurred in patients' cells that cause the disease. And we know that MDS is caused by these DNA mutations, but exactly what they were and the full sort of comprehensive look at that was not yet known. So we were using sequencing technology in that first grant to look at specific genes that were mutated in AML and other blood cancers within the MDS patient samples. But what that research allowed us to do was to also build a bank of samples that patients had generously donated so that we could carry on more extensive research and sequencing studies over the next 2 years as technology rapidly expanded. And those studies that I initiated in 2005 really formed the basis of my research career for the past 18 years. And as technology has advanced rapidly, we've been able to approach patients and use patient samples to really decipher and understand the genetic basis of this disease. So it's really incredibly valuable that we received these funds early on and that the patients were participating in the research with us to move the field forward. The sequencing studies that were initially funded by the AAMDS Foundation were really setting the stage for addressing this question of, we knew that MDS was caused by mutations in the DNA of the MDS cells, but we didn't understand fully how they cooperated between themselves, the mutations, and how they caused disease, and then how they ultimately led about a third of patients with MDS to go on to get secondary acute myeloid leukemia. And so building on the research that we did when we initially started in 2005 with studies, there was a big technology revolution in 2008, which allowed the sequencing of the entire genome. And so this is the instruction manual that is in cells. And by sequencing all of the DNA that contributes to that, we're able to understand really why these cells and what the genetic changes were. So given that, we collaborated with several people. One was Tim Grobert, who was here at Washington University at the time, and the McDonald Genome Institute that was here. And we really built off work that was pioneered by Tim Lay, Rick Wilson, and Elaine Martis that were here. And so we identified 7 patients with MDS that had gone on to develop secondary AML. And by sequencing using the entire genome, we were able to identify mutations and the changes that occurred over time in patients' bone marrow cells. And this really was key for several findings. First, we found that most MDS patients have many mutated cells in their bone marrow, not just a small minority of cells, but the majority of cells were actually mutated. And the second thing was that their mutated cells consisted of many related clones. They weren't all identical. And this was important because as people progressed from MDS to a secondary AML, they gained new mutations, or they had very rare cells that were mutated that started to grow and expand. And that's really what contributed to the progression to their secondary AML. It helped us identify, what the order of mutations were when they were acquired in the MDS cells, but it also had an impact on thinking about therapeutics. Trying to target mutations that are in cells made sense to target very early acquired mutations that were present in every cell. And then when monitoring patients that were undergoing treatment, sort of knowing all the mutations that were present in the cell was really important. And so that work has continued and started to influence some of the research that we're doing now and pushing forward with different new therapies that are available. So I was fortunate in 2012 to be awarded additional funds from AAMDS to carry out work related to one of the most common group of genes that are mutated in patients with MDS. So our group and others had discovered around that time that there were a set of genes that regulate the RNA splicing in cells. So this again is a process that all our normal cells do, but in patients with MDS that had mutations in genes that regulate this process, they had dysregulation in sort of the blueprint in how cells were interpreting the DNA. And so what we have found over the years is that those changes in those cells that occur in about 50% of patients make the cells vulnerable to do different treatments and new treatment strategies. And so what we've been working on recently is, again, through collaborative studies with Tim Grobert at Massachusetts General Hospital and Zongshen Yu here at Washington University, is different approaches and using and repurposing chemotherapy and drugs for other diseases that we can now use for MDS that are the cells that are mutated are vulnerable to these treatments. And so we have one ongoing clinical trial that's sponsored through a spore and leukemia grant funded by the NIH that is open at Massachusetts General Hospital and here at Washington University, building on these findings. And then we have additional drugs that we're testing, again, to target this vulnerability in the cells. So I think the work that we're very excited about is moving some of these new treatments into the clinic to try and improve the outcomes for patients with MDS. Yeah, the work that we do really relies on teamwork, and trainees, including pre-doctoral students and postdoctoral fellows, are a vital part of that. And so my laboratory, as well as the group that we're in here at Washington University, pays particular attention to this to try and recruit and retain young researchers in the field. We have an initiative that is being funded through the Edward P. Evans Center for MDS here at Washington University, where we fund up to 2 postdoctoral fellows each year to get training specifically in the basic research side of MDS research, but also have opportunities to shadow in the clinic. So when I see patients in the clinic that have MDS, we'll ask if they're willing to have our students and trainees come and listen and talk with them about their disease and their treatment. And so I think we take a multiple tiered approach where we have basic science lab teaching. We incorporate the latest literature that we review with the trainees. And then we also engage the patients for the clinical care and to hear how this has impacted them in their journey. And so I think with this multiple approach, it's going to be critical to help retain these researchers in the field. The work that we've done and has in the field has really relied on collaboration. And this is something for me when I was first funded by AAMDS in 2005. I was able to go to a conference that is sponsored by the foundation. And this was just very valuable to meet and interact with other researchers in the field as I was just beginning. And I think the field is going to advance, continue in the future with this collaborative work. And so for research, I think having these collaborations, continuing to bring people that are in research fields that have knowledge that could expand and enhance the MDS research field is going to be critical. And really, now that we're understanding that the MDS can develop and evolve from other diseases before a patient presents with MDS, I think a huge push in the research field is focusing on understanding that process so that we can intervene earlier, potentially prevent the initiation and start of MDS. And then really for patients to cure this disease, or as I said, prevent it and improve the quality of life that they have, and really to expand the treatment options that patients have available.